Plant tissue culture. This is a technique of growing plant cells, tissues, and organs in an artificially prepared nutrient media under aseptic conditions. Importance of plant tissue culture. A large number of plants having identical features to the parental plant can be produced by this method. Plants can be produced within a short period of time. Many plantlets can be produced without seeds. Healthy and disease-free plants can be propagated by this technique. Endangered plants can be preserved or conserved by this technique. Technique involved in plant tissue culture. Sterilization. It is the technique employed to get rid of the microbes such as bacteria, fungi in culture media, vessels and explants. Maintenance of aseptic environment. During in vitro tissue culture, maintenance of an aseptic environmental condition should be followed. That is sterilization of glasswares, forceps, scalpels and all accessories in wet steam sterilization by autoclaving at 121 degrees centigrade for 15 to 30 minutes under pressure, or dipping in 70% ethanol followed by flaming and cooling. Second one is sterilization of culture room. Floor and the walls are washed with detergents and then with 2% sodium hypochlorite or 95% ethanol. The cabinet of the laminar airflow is sterilized by clearing the work surface with 95% ethanol and then exposure of UV radiation for 50 minutes. Sterilization of nutrient media. Culture media are dispensed in glass containers plugged with non-absorbent carton or sealed with plastic closures and then sterilized using autoclave at 121 degrees centigrade for 15 to 30 minutes under pressure. The plant extracts, vitamins, amino acids and hormones are sterilized by passing through millipore filter with 0.2 mm pore diameter and then added to the sterilized culture media inside a laminar airflow chamber under sterile conditions. Sterilization of explants. The plant materials to be used for the tissue culture should be surface sterilized by first exposing the material in running tap water and then treated it in surface sterilization agents like 0.1% mercury chloride, 70% ethanol. And the next step is media preparation. Success of a tissue culture lies in the composition of the growth media, plant growth regulators and the culture conditions such as temperature, pH, light and humidity. MS nutrient media is commonly used. It has a carbon sources, vitamins, hormones. White media, nitrous media are also be used. Medium may be solid or semi-solid or liquid. For solidification, a gelling agent such as agar is used. Culture conditions. The pH of the media is normally adjusted between 5.6 to 6 for best results. Temperature. Culture should be incubated normally at a temperature of 25 plus or minus 2 degrees for optimal growth. Humidity and light intensity. Culture requires 5 to 60% relative humidity and 16 hours of photo period by illumination of cool white fluorescent tubes of approximate 1000 lux. Aeration. Aeration to the culture can be provided by shaking the flasks or tubes of liquid cultures on automatic shakers or aeration of the media by passing with filter sterilized air. Incubation of callus. Explants of 1 to 2 cm sterile segment selected from a leaf, stem, tuber or root is inoculated. Means transferring the explant to the sterile glass tube containing a nutrient media in the MS nutrient media, supplemented with auxins and incubated at 25 degrees centigrade plus or minus 2 in an alternate light and a dark period for 12 hours to induce cell division. And soon the upper surface of the explant develops into a callus. Callus is a mass of unorganized growth of plant cells or tissues in vitro culture media. Embryogenesis. The callus cells undergo differentiation and produces asomatic embryos known as embryoids. The embryoids are subcultured to produce a plantlets. Hardening. The plantlets developed in vitro requires a hardening period and so are transferred to a greenhouse are hardening chambers and then to the normal environmental conditions. Hardening is the gradual exposure of the in vitro developed plantlets in humid chambers in diffused light for accumulation so as to enable them to grow under normal field conditions. Types of plant tissue cultures. So based on the explants, they are classified as organ culture, meristem culture, protoplast culture, cell suspension culture. First one is organ culture. The culture of the embryos, anthers, ovaries, roots, shoots or other organs of the plant on a culture media. Meristem culture. The culture of any plant meristematic tissue on a culture media.